Introduction. Biochemistry plays a crucial role in understanding the intricate mechanisms and processes within our bodies. In the realm of medical sciences, a comprehensive knowledge of the biochemical pathways that regulate various aspects of human metabolism is of utmost importance. This script aims to provide a detailed exploration of 12 key topics in biochemistry that are directly relevant to medical students. By delving into the mechanisms behind insulin signaling, glucagon regulation, adiponectin and lipid metabolism, leptin signaling, growth hormone functioning, thyroid hormones, cortisol response, epinephrine release, ghrelin's role in appetite regulation, PERS and transcriptional regulation, AMP-activated protein kinase, AMPK, and cellular energy sensing, and the influence of estrogen and progesterone on energy metabolism, we will gain valuable insights into the intricate workings of our body's energy regulation system. Finally, we will conclude with a holistic synthesis of the information presented. Insulin, a hormone secreted by pancreatic beta cells, plays a crucial role in regulating glucose metabolism. Upon binding to its receptor, insulin initiates a cascade of intracellular events that promote glucose uptake, storage, and utilization. One such mechanism involves the translocation of glucose transporter proteins, predominantly GLUT4, to the plasma membrane of insulin-sensitive tissues like skeletal muscle and adipose tissue. This allows efficient glucose uptake from the bloodstream and its subsequent conversion into glycogen or fatty acids, depending on the body's energy demands. Additionally, insulin inhibits hepatic glucose output by suppressing glycogenolysis and gluconeogenesis. Dysregulation of insulin signaling can lead to metabolic disorders such as diabetes mellitus. Contrary to insulin, glucagon is secreted by pancreatic alpha cells in response to low blood glucose levels. Glucagon promotes the breakdown of glycogen stores through glycogenolysis within the liver. In a process involving the activation of adenylate cyclase and cyclic adenosine monophosphate, CAMP, glucagon stimulates protein kinase A, pKa, which, in turn, phosphorylates glycogen phosphorylase. This activation of glycogen phosphorylase triggers the release of glucose 1-phosphate from glycogen. The liberated glucose 1-phosphate subsequently undergoes further metabolism to produce glucose 6-phosphate and enter the glycolytic pathway. Through this mechanism, glucagon ensures the steady supply of glucose during extended periods of fasting or increased energy demands. Adiponectin, an adipokin secreted by adipose tissue, exerts significant influence over lipid metabolism. It promotes the oxidation of fatty acids within skeletal muscle and liver, thereby increasing energy expenditure and reducing the accumulation of lipids. Adiponectin also enhances insulin sensitivity in peripheral tissues by activating AMP-activated protein kinase, AMPK, and peroxisome proliferator activated receptor alpha, PER-alpha. By modulating the expression and activity of key enzymes involved in fatty acid oxidation, adiponectin supports the process of lipolysis, leading to decreased triglyceride levels. This hormone also exhibits anti-inflammatory properties and plays a role in preventing the development of metabolic disorders such as obesity and type 2 diabetes. Leptin, a hormone primarily secreted by adipocytes, acts as a key regulator of energy balance. It acts on the hypothalamus to inhibit appetite and increase energy expenditure. Leptin receptors, found in various regions of the brain, relay signals that regulate feeding behavior, satiety, and energy homeostasis. Increased adipose tissue mass leads to elevated leptin levels, signaling the brain to decrease food intake and increase energy utilization. Leptin deficiency or resistance can lead to obesity and leptin insensitivity, often observed in individuals with a genetic predisposition or lifestyle factors contributing to excessive weight gain. Growth hormone, GH, secreted by the anterior pituitary gland, plays a vital role in regulating protein metabolism. GH stimulates the synthesis of insulin like growth factor 1, IGF-1, in the liver, which acts as a mediator for many of GH's effects. Under the influence of GH, protein synthesis increases while protein degradation decreases, leading to an overall positive nitrogen balance. GH promotes amino acid uptake into cells and enhances their incorporation into proteins. Moreover, GH also promotes lipolysis and inhibits glucose uptake, leading to increased availability of substrates for protein synthesis. The thyroid hormones, thyroxine, T4, and triiodothyronine, T3, have a profound impact on the regulation of metabolic rate. These hormones are synthesized in the thyroid gland and play a crucial role in maintaining overall metabolism and body temperature. T3, the biologically active form, binds to nuclear receptors in target cells and increases the transcription of genes involved in energy production and utilization. Thyroid hormones ensure efficient oxygen consumption and regulate basal metabolic rate. Any imbalance in thyroid hormone levels is associated with metabolic disorders such as hypothyroidism, reduced metabolic rate, or hyperthyroidism, increased metabolic rate. Cortisol, often referred to as a stress hormone, is released by the adrenal glands in response to various stressors. It plays a critical role in mobilizing energy reserves and adapting the body to stressful situations. Cortisol stimulates gluconeogenesis, particularly in the liver, resulting in increased blood glucose levels. It accomplishes this by promoting the breakdown of muscle proteins and increasing the availability of amino acids for gluconeogenesis. Furthermore, cortisol supports lipolysis in adipose tissue, leading to the release of free fatty acids into the bloodstream. Elevated cortisol levels over prolonged periods can have detrimental effects on metabolism, contributing to conditions such as Cushing's syndrome. Epinephrine, also known as adrenaline, is a hormone released by the adrenal glands during the fight or flight response. It prepares the body for intense physical activity or stressful situations. Epinephrine increases heart rate and blood pressure, while simultaneously altering metabolic processes to provide the necessary energy for immediate action. It promotes glycogenolysis in the liver and skeletal muscle, ensuring the availability of glucose as a fuel source. Epinephrine also stimulates lipolysis in adipose tissue, releasing free fatty acids for energy production. Furthermore, it enhances oxygen and nutrient delivery to skeletal muscles by increasing blood flow. Ghrelin, primarily secreted by the stomach, is a hormone involved in appetite regulation. It plays a significant role in initiating food intake and stimulating hunger. Ghrelin acts on the hypothalamus, specifically the neuropeptide Y/agouti-related peptide, NPY/AGRP, neurons, which promote feeding behavior. It also inhibits pro-opiomelanocortin, POMC, neurons that suppress appetite. Ghrelin levels increase before meals and decrease after food intake. Any disruption in ghrelin signaling, such as alterations in ghrelin secretion or receptor function, can contribute to appetite dysregulation, leading to conditions such as obesity or anorexia. Peroxisome proliferator activated receptors, PERS, are a family of nuclear receptors that play a pivotal role in the transcriptional regulation of metabolism. They control the expression of genes involved in lipid metabolism, glucose homeostasis, and inflammatory pathways. PER-alpha regulates fatty acid oxidation in the liver, while PER-gamma promotes adipocyte differentiation and regulates glucose uptake in adipose tissue. Activation of PER-delta influences fatty acid oxidation and glucose metabolism in skeletal muscle. PERs serve as therapeutic targets for metabolic disorders such as dyslipidemia, insulin resistance, and type 2 diabetes mellitus. AMP-activated protein kinase, AMPK, is a critical energy sensor that maintains intracellular energy balance. AMPK is activated in response to a decline in ATP levels or an increase in AMP levels, indicating the need for energy generation. Once activated, AMPK phosphorylates various downstream targets involved in energy metabolism, including enzymes associated with glucose and lipid metabolism. AMPK activation promotes glucose uptake, fatty acid oxidation, and mitochondrial biogenesis, while simultaneously inhibiting energy-consuming processes such as fatty acid synthesis and protein synthesis. Dysregulation of AMPK has been implicated in diseases such as obesity, type 2 diabetes, and cancer. Estrogen and progesterone, the primary female sex hormones, play essential roles in regulating energy metabolism. Estrogen influences body fat distribution, favoring subcutaneous fat deposition over visceral fat. It also enhances insulin sensitivity and glucose uptake, contributing to improved glycemic control. Progesterone, on the other hand, promotes appetite and may impact energy expenditure. During the menstrual cycle and pregnancy, fluctuations in estrogen and progesterone levels affect metabolic processes, highlighting their intricate involvement in energy regulation. Imbalances in these hormones, as observed in conditions like polycystic ovary syndrome or menopause, can have significant implications for energy homeostasis. In this comprehensive script, we delved into 12 meticulously selected topics focusing on insulin signaling, glucagon regulation, adiponectin and lipid metabolism, leptin signaling, growth hormone functioning, thyroid hormones, cortisol response, epinephrine release, ghrelin's role in appetite regulation, PERS and transcriptional regulation, AMPK and cellular energy sensing, and the influence of estrogen and progesterone on energy metabolism. By understanding the intricate mechanisms behind these biochemical pathways, medical students can develop a holistic comprehension of human energy regulation and its implications for overall health.